This is the first patient conference that we've been able to attend for the NIGMS repository since the start of the COVID pandemic, and it was really wonderful to be here and to meet with you all. I know I recognize a lot of faces here from the blood draws yesterday, and I just can't tell you how much we appreciate it. And today I'm just going to be telling you a little bit about what we do at Coriell, what we do at the NIGMS repository at Coriell, and how all of you who so generously donated blood yesterday are contributing to biomedical research around the world. So just a brief outline of what I'm going to be covering today. First, I'm going to be talking about biobanks or biorepositories. I said that word a lot yesterday, and I just want to let everyone know exactly, what is a biobank and what is the NIGMS repository at Coriell. I'm also going to talk about kind of the reasons why you may want to consider donating a blood or tissue sample to a biobank to create cell lines and how that process works. For many of you, you already went through that process yesterday, but if there are other people that are still interested to donate in the future, I'll walk you through that. And then at the end, I'm just going to talk about some of the research uses of your sample. So if you donated blood, what happens to it? What happens to the cells that are generated from your blood sample, and what can they contribute to in the future? So first, just a little bit of background about biobanking and Coriell. So a biobank or a biorepository is a collection of biological materials used for research. And so these are typically libraries of properly stored biospecimens derived from patient samples. Okay, these can be cell lines, DNA products, they can be stem cells, they can be all different types of DNA, but they are biospecimens derived from human participants. And they are properly stored, they are high quality and well-characterized samples that undergo rigorous quality control and have associated data with them. So they are centralized and easily accessible resources for research. And one of the reasons that biobanks are so important is that they provide a reproducible aspect to research, because all samples derived from donor specimens are exactly the same. They're well characterized, they have undergone extensive quality control to ensure that every single cell line derived from your sample is exactly the same. So that if multiple researchers try to obtain cell lines from you, they know that they're getting exactly the same thing every time. And biobanks are really invaluable tools to catalyze research around the world. So at Coriell, or in the NIGMS repository, we accept whole blood, which is what a lot of people donated yesterday. We also accept skin biopsies or already established cell cultures. So if you are a patient or family who have worked with an investigator or a lab and they have already established a cell culture from your samples, we do also accept those as donations into the repository as well. And from these donated samples, we create different available products. So cell lines, we have lymphoblastoid cell lines, which are derived from blood, fibroblasts from skin, and we have DNA that we've extracted from these cell lines that we also distribute to researchers. At Coriell, we store samples in our cryogenics facility. It's a state-of-the-art facility where cells and tissues are preserved in sub-zero temperatures. So cells are stored in liquid or vapor nitrogen tanks at almost minus 200 degrees Celsius, and we have freezers where we store DNA samples at minus 80 degrees. So everything is safe and secure in our cryogenic facilities at Coriell. We have redundant backup power generators. We have off-site sample storage to ensure that there is never any true loss of sample. And in the 50 years that the NIGMS repository has been operating at Coriell, we have never experienced that. So just a bit of information about the Coriell Institute. This comes from our mission statement. We are dedicated to providing well-characterized, high-quality cell lines and DNA to stimulate and facilitate biomedical research, teaching, and the diagnosis and prevention of human genetic diseases. And so this is taken from the aspect of our mission statement that relates to the biorepositories at Coriell. So we are an independent, nonprofit research organization. So we have primary research that happens at Coriell, that's kind of one half of the Institute, and the other half is dedicated to our biorepository operations. At Coriell, we house a number of diverse and world-renowned biobank collections. We have several NIH-funded biobanks, the NIGMS repository being one of those, and a number of private biobanks as well. And they cover a number of different topics. Some, like the NIGMS repository, are disease-specific. Some have to do with human populations around the world, and some even have non-human species as well. The NIGMS repository at Coriell is funded, as Danae mentioned, by the National Institute of General Medical Sciences, which is one of the institutes of the NIH. It has been housed at Coriell and continuously funded since 1972, so since before I was born, which dates me, but that's life. It contains almost 12,000 different cell lines, almost 6,000 DNA samples, and almost 100 different induced pluripotent stem cell lines. And we represent over 1,100 different genetic disorders in the repository. So since the 1970s, we have been collecting samples from a number of patients representing a number of different genetic disorders. These include heritable and non-heritable genetic disorders, chromosomal abnormalities. We also accept samples from apparently healthy individuals. A number of the parents donated yesterday. So even if you're a carrier or not a carrier, we also accept samples from apparently healthy related individuals, individuals from diverse geographic origins, and also stem cells. We have a global distribution, so we distribute cell lines and DNA to scientists around the world. Last year, we distributed samples to 70 countries for research purposes, and to date, NIGMS samples have been cited in over 7,000 scientific publications. So why donate your sample to a biobank? Well, one of the reasons that we exist is to enable research around the world. So scientists need access to rare samples or populations in order to advance their research. And one of the ways to facilitate this is, instead of every single individual scientist going out to collect and recruit samples from patient populations themselves, they can come to one place, one resource, where we have collected all of this and characterized it all for them. So in using biobanks, investigators and researchers have quick and affordable access to obtain samples. They are guaranteed high quality and well-characterized samples, and we are able to distribute samples all across the world to researchers, so not just in the United States. Now, who is eligible to donate samples? Many of you yesterday already participated in our blood draws, which is wonderful, but we accept samples from any individual diagnosed with a genetic disorder or chromosomal abnormality, and clinical documentation is required for us to get your sample from the labs into our catalog. And so I know we'll be following up with a lot of you who donated yesterday to obtain as much of this documentation as possible. We accept samples, as I mentioned, from affected individuals and family relatives. We accept blood, skin, and established cell lines, as I mentioned. And I also would like to highlight this last point here, because it's very important, is that one of the things that we are really trying to do a better job of and really actively engaged in is to recruit, do a better job of representing global diversity in our collections. And so we encourage diversity in sample collections to ensure that there is equal access and equal representation among populations for all of the samples in the repository. And this especially relates to historically underrepresented communities in biomedical research. So for those of you who may still be interested in donating and didn't get an opportunity to join us yesterday, there is still time to do that. So we do remote, let's say, sample collection. So all you have to do is reach out to us. Our email, I'll have this up at the end, is nigms@coriell.org. So nigms@coriell.org. You can just email us, let us know you're interested, and one of our project managers will reach out to you and walk you through the entire process. All you have to do is email us, request a sample collection kit. We will mail it to you with forms to complete. You have your blood drawn or tissue biopsy taken by your doctor, and then ship the kit back to us. So everything is prepaid. There is no charge to participating. In fact, we do reimburse up to $40 for blood draws if you have incidental charges like a copay. for visiting your doctor to have the blood drawn. So all of this is at no cost to the donors, and we will walk through each and every individual family to answer any questions that they have or provide any advice that we can to make the process as easy as possible. We also have a donor portal, which really is a very thorough step-by-step guide through the entire process on our website. So if you're interested and you email us, we will give you a link where you can access this. It has a guide of how to best contact your doctors, how to pack up the sample kits, and answers to lots of frequently asked questions, as well as all the documentation that you would need to fill out the kits. So some of you also may be wondering, if you donated samples yesterday, what is going to happen to the blood that I gave you? So we'll collect samples, we'll bring them back to our facilities in Camden, New Jersey, and process the samples to create cell lines in our labs. Once the cell lines are established and quality controlled, and we have all of the necessary clinical documentation that we need, we include samples into our online catalog where researchers can go to. They can look for homocystinuria samples if they're interested in researching HCU, and order them from our catalog. We ship and distribute samples to researchers and then report all of this back to the NIH on a yearly basis. And so now some of the different research uses for your samples. So when your sample leaves the biobank, what happens to it? It can be used in a variety of different ways, from basic research into human biology, the identification of new genes, the development of disease treatments, and validation and proficiency testing. So there is really no limit to the types of research that can be done using your samples. These types of cell lines are also invaluable for use as reference materials. So especially now, where a lot of people have ready access to their genetic test reports, you have characterized mutation data for any mutations that you may harbor or be a carrier for. These samples can be used to improve the accuracy of diagnostic testing, because labs can use them as reproducible tools to validate their clinical testing. So we can improve the diagnostic criteria that is used to then further diagnose patients in the future. I also just wanted to highlight some of the successful collaborations that we've recently had. So one is with the Chordemia Research Foundation. We began working with them in 2016, and we're working with them to develop cell lines for collection at Coriell. We have a wonderful working relationship with them and a really successful collaboration. And recently, their foundation, along with investigators that they're working with, developed 4 different gene therapies as a direct result of work that they did using cell lines that they established at the NIGMS repository, and that was very exciting. Some of the more interesting things that could happen, we have worked with the Fight Niemann-Pick Foundation for quite a number of years to develop a resource for NPC1. And one of the things that really kind of surprised us was there was a researcher at Columbia in New York City who was studying Ebola and used cell lines established at Coriell to come up with a link between the NPC1 gene and the Ebola virus. And this was back during the height of the Ebola outbreak in 2014 and 2015, where they discovered this link as a possible cure for the Ebola virus moving forward, which is really, really exciting. So I just wanted to take this opportunity to thank each and every one of you who donated yesterday, and just if there are any more people who are still interested in donating to the repository, please reach out to us. We do have a pretty good representation of HCU and acidemia samples in the repository, but many of these samples are older and do not have mutation confirmation data associated with them. So one of the things we're really excited about working with all of you is to have newer samples that have better data characterization alongside them, and to hopefully recruit as many samples from historically underrepresented communities as possible. So with that, I will say thank you very much. Our email again here, nigms@coriell.org, and thank you for having us.
Danae, can I say something? Yeah. So, Hi, I'm Kim Chapman. So I want to give a real life example for you all for your disorders, because he gave some different disorders. So the PA Foundation funded me in, is Jill here, 2012, 2013, something like that. And what we did is we used the Coriell PA lymphoblastoid cell lines and did expression profiling, and we're able to identify the expressions in that were different in individuals with PA, their parents, and then a control group. So I want to say that we actually already use some of these cell lines. We use some of the old ones. It would be nice for some of the new genotyping ones, because I actually had to sequence the PCCA and PCCB genes in those so that I could characterize my population. The other thing is that what was really essential with that particular study was the parents, so the carrier parents, because it allows you to ask different questions. And so I also encourage parents, if your child is donating, to also donate, because that actually really, really, really helps us when we're doing analysis. Sorry.
No,
I thought I would use a real example out of this group that was actually funded by PA Foundation. It's now 10 years ago, but.
That's fantastic. And yes, thank you. Thank you very much.